Will, if you can unmute yourself and turn on your cameras, we'll get started. Wonderful. Thank you both. Okay. Thank you everyone for joining us today for the Hatfield Marine Science Center Research Seminar. My name is Cinnamon and I am the Research Program Manager at OSU's Hatfield Marine Science Center, and I will be your host today. We have a couple of logistics to talk about and a couple of announcements, and then I'll hand it off to today's speaker. I just wanted to let everybody know that their mics, cameras, and screen shares are turned off. If you could help us out by keeping them that way throughout this talk, that would be great. We do encourage you to ask us lots of questions, but please use the chat box and we'll get to those at the end. I also wanted to let folks know that we will be recording this session, and I am putting in the chat box right now where you can watch that at a later date or share that with others that are unable to join us today. So a couple of announcements. I just wanted to put in a plug for next week's seminar, which is already July 16th. We're halfway through the month. Can you believe that? Next week we have Dr. Athena Rissick as Assistant Professor of Biology and Marine Science in the New College of Florida. And she's going to be talking about the Floridine manatee's hearing and boat collision and talking to us a little bit about an integration of laboratory and field studies. So if you're interested in that, please join us, the same place, same channel. Just to let folks know, if you want to know more about upcoming events, you can check out HMSC's website, scroll to the bottom, and our calendar of events is there. But for today, I wanted to hand it off to Will, who invited our speaker. So Will, if you want to take it away, that would be great.
Great, thanks, Cinnamon. And hi, everybody. It's my pleasure to introduce Fabio Caltabellotta. He is a researcher who studies a lot of different things. Today, he'll be talking to us about the life history of elasmobranch fishes, so sharks and rays. But Fabio is broadly a fisheries biologist, and he does a lot of work with how we can use life history information to manage data-deficient or data-poor species, as well as doing a lot of work with management strategy evaluation, which essentially is asking, are we managing things in a way that's going to lead to good outcomes? He started his career in Sao Paulo, Brazil, where he obtained a master's at the University of Sao Paulo and then a PhD in zoology from Sao Paulo State, and then was an assistant professor at University of Santa Monserrat in Brazil, where he taught classes in fish biology and fisheries science. He then came to the United States and has been a postdoc at the University of Florida, working with Robert Ahrens to develop management strategy evaluation techniques for Gulf of Mexico red snapper. And that work is just wrapped up, and you may have seen an email went around a few months ago with the product of that research, which is this really cool web interface that uses our Shiny technology to make the management strategy tool accessible to non-fishery scientists. So it's a really great exercise in science communication and using technology to do that in an effective way. Now, having wrapped up that work, he's actually a postdoc at Oregon State here in my group in Coombs, and his work is going to be jointly with Oregon Department of Fisheries and Wildlife. So the retirement of David Sampson last year sort of left a gap in ODFW's capacity to interact with fishery management and the Pacific Fishery Management Council. And so part of Fabio's position will be to do that scientific support of helping ODFW work with fishery management at that regional level, as well as doing some research on fisheries management science. Of course, we hired Fabio right at the beginning of this whole COVID thing, and so he is still at his home in Gainesville, Florida, where it's much hotter than it is here. And he hasn't been able to travel to Oregon yet, but hopefully he will someday when we are all in better times. But for now, I hope we can hear all about some cool elasmobranchs.
All right, Fabio, I think you can take it away.
Yeah, thank you so much, Simon. And thanks so much, Will, for introducing myself. So yeah, today I'm just going to talk a little bit about one of my particular projects, the life history of elasmobranchs, the elasmobranch species. That's a personal project that, like, I have a really passion for sharks and rays that I have been working on since my master's. And one of my main aims is to advance the assessment of data-deficient and the conservation priority of the sharks and rays species. So I just started at the beginning, right? So during my master's in Brazil, I started working on with some blue shark embryos. So I have some cool ideas about, let's try to check if we can find some kind of bands in sharks, in angler sharks. And I remember that time that I just email like for Greg Kaye, Stevie Campana, and ask about, oh, I'm doing that. And I was new in this science, the aging growth stuff science. And I asked about, hey, I can do something with this vertebrate or not? And like the answer is, no, it's difficult to find some kind of band deposition in an embryo. At least you have some kind of pre-birth bands in some sharks, but you can try to do something. So I have some kind of cool ideas, like let's get the vertebra from the embryos, like do the same thing that we have in the elasmobranch from the FAO book that tells you all the methodology that you need to do to work on aging growth stuff for elasmobranch. So I did the section, and then I see, oh, there is no band here. So what I can do? So came into my mind, like we have the back calc, right? When we have the age of fishing, once you know the other's length age. And what I can do using this vertebra. So I know that there is some kind of information in the literature about when the blue shark embryo born around 45, 50 centimeters, and the gestation time took around 12 months, like 1 year. So I just used all that information and did some kind of back calc to get a roughly idea about the age in months. And what I get this Gompertz model here, right? That is really simple. And as we can see, like between the 4 and 5 months of gestation, you are near the inflection point of the Gompertz growth curve. And that means that during this time, we have a change in the growth. But what surprised me is that Lega and Vore in 97, they found out that during this size range, between the 4 and 5 months of gestation, you have a transition between the yolk to the maternal nutrition. And that makes totally sense for me. So everything started here during my master, right? So then I started my PhD. After finish that, I started my PhD in Brazil again, and working on the southeast coast of Brazil. That's the place that I'm talking about. I work with three species of guitarfish. One is Pseudobatus horkali, that was Rhinobatus horkali, just changed the genus. The Brazilian guitarfish, that's critically endangered. Another one, Pseudobatus persellens, that was Rhinobatus persellens as well. The southern guitarfish, sometimes called a guitarfish, that is near threatened by UCN. And Zapteryx brevihostris, the short-nosed guitarfish, that's vulnerable by UCN. So all these species, they are lecitotrophic and exhibit like horkali, size about 140 centimeters of total length, persellens 100, and Zapteryx brevihostris around 65 centimeters of total length. And although these species, they are not targeted by the demersal fisher that we have here in this space in Brazil, even by small to large scale commercial fishers there. We have like artisanal fisher, commercial fisher, all types of fisher there. But there is some kind of fisher there, like the part-trolling fisher, that gets a lot of these species. So 2 of these species, they are recognized in Brazil that critically endangered there. And you can't catch these species over there. But unfortunately, this is still happening. It's really hard to control this kind of fisher. So it's important to draw attention that 36% of these guitarfish species, they are threatened. Like 2% are critically endangered, 5% endangered, and we have like 29% vulnerable in the world because of their coastal habitats and you have a lot of interaction with fishers. Some of them are endemic and the medium large size, and there is a really high economic value for some of that. And therefore, the growth parameter estimation, it's really important. It's crucial for assessing their vulnerability to the fishing pressure. So, okay, so what I did, I just submitted a paper, and as part of the scientific process, we received back the first round of questions from the reviewers. So we sent out, we have done a lot of people have working on like the same methods, the same thing, which is applying different growth models, multimodal inference, the MMI, and we run these issues here. Like one of these issues that talks about the sample size that we have. Of course, we have for the Pseudobaetis horkali around 148 species, individuals. We don't have more than that. And then another issue talking about the L0, because use the L0, we fix these values, and they are trying to, why are you doing that? But yeah, but for the sample side, there is another thing. There is like a kind of threshold established in the literature, like of 200 individuals. So there is a lot of things that people, they are like some rules of thumbs, right? I need to follow this and that's okay. You have the receipt, just follow the receipt. You can have your study done, right? So I did that, but I received that back. So what do we need to do? What we can do, right? So what we did. So we did something that allowed us to overcome these hurdles. So instead of taking all the standard frequentist approach, right? We like, hold on, let's thinking about how can we overcome these hurdles. And as such, like we decided to implement a kind of Bayesian version of the 3 parameters von Bertalanffy curve. And this allowed us to deal with this uncertainty result for the low sample size, bound the parameter estimates that can be freely provided in the Bayesian framework, and joint distribution of the parameters in simple during the HMSC sampling, right? So all of these motivated us to take a kind of departure of what people normally do. They run just the NLS, the nonlinear least squares, to fit the data and move on. Okay, it's fine. But we know that these parameters, they are highly correlated, right? So we want to be able to estimate this joint posteriors, things like that. And again, we want to be explicit about like the sample size. That's really important, right? So what's the advantage of the Bayesian approach? Like for me, it's a new approach because I was like, okay, you can just use all this frequencies approach and you can get all this aging growth stuff done, right? But we find some cool things about what's the advantage of using this Bayesian approach. So as a result of that, we don't need to have like correction to deal with the adjustments of the interval estimation as in the frequencies framework, which rely on asymptotic normality, right? Also, this resulting in less out of bounds issues that because of the estimation of the Hessian matrix in the frequency settings, and which given us like the ubiquity of strong correlation between the von Bertalanffy growth parameters. That is important to not artificially inflate the interval of the uncertainty. But we recognize that moving on to the Bayesian framework is not accurate at all. So like for the sample size, etc. But we got this paper published. So yeah, works. So we got out there, and because we took this kind of different approach that normally people do, at least in the shark and ray Asian world, and come across to the residual comments, and this motivated us to kind of thinking about to continue on this framework, right? Which seems to solve a quite a few the issues that comes up in the growth model in literature. So this is like is really what we have for the elasmobranch, right? So elasmobranch is one of the biggest problem is the data deficient. So I just update a little bit. We have today about 1,124 species assessed, and the number of data deficient is like 438 species. So in elasmobranch, data deficient is a huge issue. And we're talking about like 40%, not more 45, because some of them move around the other categories, right? And so how can we make an estimate? So what we can do, like maybe to the IUCN status, maybe just interested in the population trend, maybe in local prediction, maybe in a management strategy. And we need some more information for these data deficient species, right? To be able to kind of do some things with the rest, because we have around 60% that we have some other categories, but we have at least 40% that we don't know anything about. And we are not still like talking about species that we know a ton of that for the 60, right? There is really not a lot of elasmobranch species that we know. Even for the species that are not data deficient. Like for instance, like here, we have Isurus paucus, right? There is no growth curve for it, just brought for the Isurus oxyrhynchus. So it's considered endangered at the IUCN, the global level, but I think that's the Mediterranean, it's considered as data deficient. And here's the Narcine brasiliensis that we have been working on, the data deficient, right? So even for some of the species that IUCN is considered not data deficient, we still don't have information about life history. And here's a good example. We have a bunch of carcharinated sharks, right? That may be like this. We have 54 species in the Carcharinae family, and at least 5 species, we have like multiple growth curves. Like here, the best example, like plumbeos, Carcharinus plumbeos, Limbeus, Paranacidoca, Carcharinus brevipina, and Spirina lewini. That we have some data, a good data, right? But for the most of part, we have just one growth curve or no growth curves, right? So why do we care? This is really important. Why do we care with fill in all these data deficient species? Like life history information, it's really important for all stock assessment models, right? So this needs to be dumped here. Or at the same time, to be here to inform some age length compositions, right? So I just highlight the growth because it's really important. So even if for the age-based models or length-based models, we need to have this information. It's really important. And at the same time, we need to be aware about all the process errors, all the bias that goes into that models that come from the growth literature. So this slide is really interesting because this is just the best practice, right? The use of LNOT that Dr. Kaye, one of my collaborators, and you need to use the LNOT because LNOT, it's something that is data, it's sized bird, not a T-naught, right? And what we have been seeing in a lot of papers that people, they are citing the von Bertalanffy with the LNOT, that's the original one, as Fabian 65, right? And then decided the von Bertalanffy with T-naught as the original one, and it's not the original one. It's the parameterization of the Beverton. And you can see in a lot of papers. So here we have some kind of source of variation that we have in this world of the Asian growth stuff. So we can look at what the relative influence of the preparation techniques. So whole vertebrate standing methods, you can use like alizarin head, you can use the ocean blue and silver nitrate, and section, have different section to do X-ray, right? And at the same time, We also have the reader accuracy and precision, the bias. And there is some models like the VGBM with random error, random effects error, that deals with this intra and inter-reader bias. And what makes us to move around this Bayesian framework is that this Bayesian model can handle those kind of problems like the sample size. That's really important. So we got accepted this paper recently that deal with a data-deficient species, Narcine brasiliensis. So Narcine brasiliensis, this is a primarily coastal and estuarine species in Brazil, found in mud and fine sand. It's endemic from the southeast Brazil to northern Argentina. And this is a really small licitotrophic species that reach maximum size about 49 centimeters of total length for females. And despite of the massive range, we didn't know nothing about. So there's some ideas how we can fill these gaps, right? We don't have information. This species is still caught by the other fishermen. So what we can do? They are really commonly caught as bycatch in Brazil. So first, that I need to be really clear, all right? So first off, like the sampling is totally opportunistic because it's from bycatch. So I have no control over it. And we have received a lot of comments from the other paper, at the beginning from the other paper, from the guitarfish about that. And this is what the majority of elasmobranch studies, it's the case. It's totally opportunistically. We don't have control about this. And they are getting like this kind of, it's not an unusual phenomenon. And the other thing we have is the bias in length data. So if you look at the histogram here, down the left, right, we have missing some small fish, and at the same time, small fish here, and at the same time, some large fish, right? That is we don't have some males, extra large males. But we already know that from a lot of different papers that that's normal, right? This kind of information sometimes you don't have because of your selectivity. Yeah, could be. So what's going on? So then because of this look like a bias, but the fact of matter is this, the male don't get that big, because this could be like, no, males just get this big and stop to grow. So in fact, is what that figure shows. It's not like a kind of selective issue. They grow totally different from the females. So what we did, so this is important to bring what we did for all this stuff. So the difference between the model that we have before for the 3 guitarfish parameters and we have now we have a model with L0. That's the original parameterization of the Wumbertal function. So we are seeing that the data is log normally distributed, right? But what's interesting here is we have another likelihood. So we have 2 likelihoods. This likelihood here, where is that saying that's the observation PI, is the observation of the bird size, is also log normally distributed around the L0, which actually estimated as part of the other likelihood. So in this way, we are taking one parameter estimation and constraining how possible the other values of the L0 by observation of L0. So this is the cool thing here. So, and at the same time, that's really important to highlight that no one has done that before, at least in the elasmobranch aging growth studies. And it's a really easy extension about what we can do. So here, what we have like a prior that makes this possibility availability of the L0, and you are actually using the real data to constrain the posterior of L0. So we take the data and constrain that to the posterior of the L0. So we are not the first one that know that we should include this information, as I showed before, right? So Kaye always like, you need to use the L0. That's information. right? But what's typically done is like you're fixing the L naught at the mean. Just fix the L naught. And because you're not include extra information about L naught, there is a lot of variance there. So just to highlight what we did, so we know that NLS is not a good choice, and we know that it's a parameter that is super correlated, all the group parameters. So why do not explicit like in the Bayesian framework that we can deal with that? At the same time, in NLS, we are assuming that error is symmetric, and we know that generally is not the case in the Humboldt growth, right? So we assume that's log normally distributed to account for all the long tail behavior that occurs. That's normal, right? And we are interested in fixing L naught or even estimating the T naught, because T naught is a kind of not meaningful biological parameter. And instead of that, we just include L naught as data, right? So the observations of L naught, they are totally incorporated in the variance in the L naught into their estimation in the model. So we do all this integration. And here, just to show you guys that we don't have any kind of difference between sex for the two kinds of measurements that we have, the disk width and total length for the length-weight relationship. We have some kind of difference here for the growth, because we know that females grow larger than males, and for both, for disk width and total length, that makes sense, because female need to have space to have all these embryos inside. And for the disk wide residuals, they are symmetrically distributed. We don't have any kind of trend. But for the length, we have that. Like we have some kind of large males, like appear falling two groups. We have one group here that's still growing and one over here that stopped to grow, right? And also here, we have the mortality at age that was similar between sex and like for the disk width and total length, with a negative exponential relationship between age and mortality. So then, after finish this work, we just got this paper accepted this week now. I think that on Tuesday, right? Yeah, on Tuesday. That's about Mitsukurino's one, the goblin shark. And this is a really cool paper and like a really cool species that I'm really happy to have like the opportunity to do such kind of work with that. So now let's talk about the Mitsukurino. And one of the best, for me, is one of the best examples that we have for data-deficient species. Even in IUCN, you're considered as less concerned, right? But it's not, we don't have information, any kind of life history information. And is the sole extinct member of the family Mitsukurinae, and one of the largest deep water sharks that we have, and lives in really deep water. Like they have a circumglobal distribution and 1,000, 1,500 meters, and maybe like can get a total length, the maximum size about 410, and maybe there is some other studies that tell that there is more, but we don't believe that. And there is only, I think that there is only we have only 200 goblin sharks caught in the entire world, and mostly are in museum collections. And you know that you can go to in a museum, just grab all the vertebrate stuff there, and oh, let me destroy that to do some kind of work. And it's considered, so as I said, like one of the least known deep water elasmobranchs in the world. And for this deep water shark, or for any other elasmobranch species that lives in deep water, getting age information is incredibly difficult and requires some kind of specialized techniques to do that. For some sharks, when you have spine, use spine, and vertebra, you try a lot of different techniques for standing the vertebra. And one of the main reasons of that is because as the central is not calcified, so the hydroxyapatite is not fully calcified, and it's really jelly. So if you try to slice as the way that we normally do, you're gonna be in trouble. You can't do that. It's really hard. So we have only one individual. That's one male that was caught in Rio de Janeiro, right, by a Spanish other trawl fisher that targets the deep water scarlet shrimp. And the question is, can we do something with just one? Like, I have more than one vertebrate, right? But just one individual, what we can do? Just, okay, describe a species, because this species was the first one, the biggest record in Brazil, I think, caught in Brazil. And okay, we start to think that, oh, we have the Bayesian method that we have been working on all this time. And let's try to include some of the information that we have, pair it with other information or bad guess or priors, and try to be able to at least to get some hopefully idea about how Mitsukurina stone is growing. Let's try to do that. So it's important to highlight that we try different kinds of bow tie sections, that something that we normally do for the species, but we are unsuccessful because of the poor mineralization of the intermediate. So we have the corpus calcareum, in fact we have the corpus calcareum, and between the corpus calcareum, the intermediate. And for this species, we don't have intermediate. It's totally jello. So yeah, so what do we need to do? Like, we have a lot of issues try to tackle and figure out what we can do to try to age that species. So I have a colleague in the lab during my PhD that he was working with another species and try to find to look at some structures using the Alcyon blue method to distinguish what is cartilage and what's not cartilage. And I have this cool idea when I look, oh, you can figure, so I can try to use that for the Mitsukurina stony. Why not? But at the same time, we need to find another approach to section this vertebrate. So then we approach it like a different kind of quadrant sections, and which quadrants we stain it with the Alcyon blue method. And at the end of the process, at the end of the staining, one quadrant that we have here, and we have all the that we assume that are the bands. So I have here the birthmark. Here you have some kind of angle change and a really angle change, right? And it's really cool because all of this work began in the past, and it just took back, I think, at the end of the last year, and when we found, oh, we have this base sample, so let's use that for this chart, right? And again, what we did, so we went out and we have some prime information about the LNOP, like from the smallest free swimming individuals. So I have two that is here in the data, two small swimming individual, and one from Iano, 2007, and another one from Castro, 2011, right? And at the same time, we include the information that we have already, like all the information, the data that we have, all the same structure that we did for the Narcina, we did for the Mitsukurina. But the other thing that we did that is different that we did before for the Narcini, use the back calculation, right? So we have only one individual, right? So we did a Fraser-Lee back calculation to get approximate information about the size at age. But another thing that's different from what we normally do when we use all this back calc, right? We have this kind of error bars here, right? Along all the data, because this is something that's really important to pass all the information about the uncertainty that we have. We are not fixing the data, right? So we brought in all the same data that we had before. So the L0, we also have some extra information about the biggest individuals that are here that ever been caught. So we have like for every single hacker, every single large Mitsukurina in the world, we have the information. I have like all the information that we have in the world about Mitsukurina stuff. And so the other thing that we did, that just because we have only one individual, like as I said, like the back calculation, right? But different that the people do, we just assume as the data as a kind of latent state model, right? That we estimate. So here we just include all the L0 data. Here we have all the Lmax data. Over here is the back calculation. And here we incorporated this uncertainty from the back calculated age in the model by conduct a kind of phraseback within the model. And this is really interesting because like the Fraser Lee assumes that L0, and so like because of the L0 is in the model, you are just estimating L0 as part of the model. So we have some data on L0, right? And we don't know L0 for sure because it's not a fix. Okay, you need to bird with 30 centimeters. No, it doesn't work like that. And we know that, right? We need to have all this kind of variation. We can fix this kind of parameter. And so a cool thing that we did, we like brought all the data and then run the model over all the L0s, and we have a posterior, and this posterior gives us back a posterior of back calc, right? And so we can deal with all this uncertainty around that. So that is because I told before that we have these error bars. And the cool thing that we have been working, because we can advance what this kind of other works I've been doing, right? So they assume that L0 is no. And we know that L0 is not no, right? We know that. So for this particular model, we bring all the increment data that we have. We bring length capture data, and just we do the backup in the model. So we have this L hat here, right, in the VB. And the difference with the other model that is no, right, and this is the case, the data, is the back calculated data and the L hat give us our best estimate about from the von Bertalanff growth function. So you are also able to get some cool parameters, not only this, not only the size, right, and the L0 and the key and the L infinity, but we also can get some other cool parameters as age at maturity and mortality at age, longevity, as we did for the Narcine brasiliensis using some life history invariants. But we have some other things that we want to do. Instead of using this life history invariants, we want to bring that to the model, like to do everything inside the model, right? So I just bring again all the search of variation. And what I want to bring attention is about adding these other two sources of variation. How we fit the model and the multi-model inference, the MMI. So it's a kind of rule to use like different models and then use the AIC and then test what's the best model to do this and what can describe the growth. But what people never been doing is try to understand how can you serve some kind of this bias that the multi-model inference bring us. Like we need to understand that, because how this kind of thing can look at your data and know that, no, this model is the best model for Carcharinus plundus, and this model is the best model for Prionaceae glauca. Doesn't work like that. And here's a good example. So I want to draw attention here. It's a paper published in a really good journal. And I have like, I just highlighted here, and I have like this kind of stuff like, do we think that this kind of species grow negatively? Because if you look like the standard error, plus minus 0.02, we're gonna have a negative key, that's impossible, and at the same time for an infinity. So the SEs that cross zeros like this doesn't make sense. So this kind of things that we know that should be positively bounded, and this publish and bring a lot of attention about the use of this kind of source and the rows of thumb and this a lot of different models. And so over here is a really good example, like for the multi-model inference. So where you're fitting a bunch of different models, sometimes very different from the actual number of parameters that we have in there. And at the same time, we don't have a really good idea about what's the species, the other life history of the species. We don't know anything about that. They're just applying this kind of models. So MMI is like, it's really interesting. And we need to understand how like we generate data on each of these growth models can be understand through this kind of approach. So how can we be able to detect the best appropriated model, right? So that's why MMI is like people are using, like raise information criteria and compare models. And the context of the elasmobranch work, we know that is not the best appropriate approach to use. And like here, I have a really good example, right? Like for my blue shark animals. Like Gompertz describe perfectly what happened in the utero, right? But do you think that makes sense? Use Gompertz for a laminiform, for a Carcharodontidae, after they're born, like they grow exponentially? Like, no way. So this kind of approach, we need to be really careful, right? Like this model, they have a lot of implicit assumptions of the biology, and instead of like, we need to ask the question if like it's biological, is reasonable to use that. So the model selection process that is the most parsimonious to the data, is that what you need to use? Like let's the AC tell me how sharks grows. And so yeah, I don't think that we need to assume that. We need to be really cautious about this. And I just get at the end of my talk. And one thing that's I saw and talk about that with Will and Karen Brab and Meg Summer from the ODFW. And it's about application of this existing methods that we have been working on in some other species like the Pacific spiny dogfish. It's called suckley, right? And I think that we can apply the exact thing that we have for this other species. So to do this, like for a species that's a priority management concern, as the Pacific spiny dogfish, it's really important because the key value is extremely low. And I personally have some kinds of concerns about how the model was estimated. And we probably have a lot of extra information, like from the car. So why we not just dump this data in the model framework that we already have and build more kind of robust estimates about what we think that's going on. And like in particular, like for this kind of Pacific spiny dogfish, they are aged just using the spines, right? And we think that we can have like the spines and vertebra, and kind of estimate some kind of correction factor between spines and vertebra, like just inside the model, and build some better estimates. So what about the age should be, and this is gonna probably bring us more about the best growth models and parameters that we can have from that. And also another thing that's like, it's one of the interesting things that we have been working on sharks as a whole. Like, we are trying to fill all these data deficient species. So we are trying to do some kind of relational model, right? Some kind of relation information that we can bring some kind of traits, phylogeny, body shape, and other information. So let's say here we have this matrix, right? So we have this axis that is like the species that we have. So in this case, we have 6 species and 3 parameters, like here, that could be the growth parameters. And there is some underlying patterns or covariance, right, between both species. And maybe that could be like phylogeny, as I said, traits, size at birth, but we also have this kind of covariance, like between the parameters. So we can try to estimate those kind of data directly, then we observe it, but at the same time can also bring these things as priors. And we know that K and length, they are negatively correlated, right? So we can bring these prior, but we can estimate it directly from the data if we have the data. But for instance, for some of particular species that we haven't observed that information yet, or we are not able to estimate that, that is in the gray boxes here, because we have tons of species that we know anything about. And we want to fill in this gray boxes, right? So what we are doing now, we are doing this for Carcharodontidae and Sphaeronidae. So we have probably 30 species along this group that we have some decent data, at least one growth curve, and some other ones, like as I showed before, like for some species, maybe 4 or 5. Yeah, 5, that we have multiple growth models. And the idea here that we are just doing right now, we are trying to estimate and fill all these holes for these species that we had nothing, because we want to fill this data deficient species, right? So you have a bunch of kind of endemic Carcharodontidae in some kind of Indonesia, for example, that's like in a particular area. And we can also like correct that for other species that we have a ton of data, and can correct the data if it's not right, because we have all this Bayesian framework that deal with the sample bias. And I think that all this kind of relational model, there is also like a good kind of analogy structure that I see that we can like do here in the Northwest, like for the rockfish, for the sebaceous, right? Because we have a bunch of species that we know quite a bit, and at the same time we have species that we know like is in the middle, and other species that we don't know anything. So the idea that is like we can take some information from phylogeny, traits, habitat types, and we can bring all together and try to make like our best guess for this species. And we know that even the Bayesian framework is not accurate at all, and we know that we still need to get data. We still need to get data from surveys and from other parts, right? And it's not a substitution for that. And just almost finishing here, and that I need to bring, we have a package that called Fish Life, and this package did not include the phylogeny, just taxonomic rank, right? As the relational information to bring information. And at the same time, I think that it doesn't deal with necessarily with all the covariance that we have from this species. And bring data from fish base. And we know that there's some data in the fish base that are not reliable at all or not has been updated. So we are doing all this kind of literature research, all this process, bring all the information that we have tons of papers, information about this Carcharinidae and Stranidae families. So finally, I just want to thank you all for your attention, and at the same time, show that we are trying to figure out some ways to improve this growth models, right? The growth curve in general. And at the same time, to fill this data deficient species, like both directly, the case of Narcini, or directly, indirectly, in the case of Mitsukurina, and kind of this large relational ways that we have been working on to fill all the data deficiencies that we are able to fill in. And I also want to thank all my collaborators, institutions, and all the people that I have been working on. And there is a lot of other people like technicians. And thank you so much for your attention and questions.
All right, everyone. If you have any questions, go ahead and type them into the chat box now. Fabio, thank you so much for spending time with us. When you were talking about data deficient species, yeah, that's data deficient species. When you only have one sample or just a few, that kind of brings things into light. I'm not seeing a lot of questions coming in, so I'm just going to give folks just a few minutes. I was thinking maybe you could talk while we're waiting for folks a little bit more about the work that you're hoping to do when you're here. Kind of some of those target species that you're looking at, maybe filling in some of that,
yeah,
those gaps.
Yeah, like as I'm saying, like I think that is a good way to. There's a lot of rockfish species that I believe that we can fill the data, right? And why not doing that if we have this kind of approach? And that's really interesting for the, like, particular for the rockfish, is because we have a different kind of reproduction, right? We have a larva that's born with between 5 to 9 millimeters. So we have size at birth. So why not using that? And I believe that we can do that. I envision how to do that. It's something that is very important. And at the same time, like for the other species, like the elasmobranch species, even if we have a bunch of information and we have like growth curves that are published, why we are not just trying to use this Bayesian approach, try to get this best estimate, this uncertainty, try to estimate the uncertainty, because, okay, we have a bunch of species that we have information about growth, right? But then when we just dump that to the models, to the stock assessment, even for the age-based or length-based, like for example, a good example, the length-based models. So you are just borrowing information from outside. And I don't think that is a really good idea to do that. Oh, there is a question here. So are there opportunities for collecting genetic samples to verify species and look for stock structure? Yeah, for sure. Yeah, like this, like I can tell, like from Brazil, we have a really big group over there working with all these genetic samples there, and not only there, but people like from there working with people here in the US, and they're doing a lot of that. Yeah, it's really important, because it's a really good question, because I'm pretty sure that we not only the stock structure, but we have a lot of cryptic species between that we don't know anything. That, oh, this is a Spirina lewini. Yeah, it's a lewini, but no, it's not a lewini. Even for the guitarfish, we have the same problem. And unfortunately, we still like waiting more information from genetic and other kind of taxonomic studies that maybe we have one more species. like between lentiginosos that we have here in US and the persimmons. We have another one that we need to figure out. And that just is a good point to bring. I don't understand why we still don't have information for lentiginosos, for Pseudobaetis lentiginosos here in US, because all the guitarfish, they're really, really endangered, threatened. So we need to be aware about that. So yeah, thanks for the question.
Thank you, Scott. It looks like we don't have any other questions. So at this time, I just want to say thank you so much, Fabio, for joining us. And like I said at the beginning, we did record today's session, so we'll put that online and folks can share that for folks that weren't able to join us today. And everybody online that's still with us, hopefully we'll see you next week. Thank you for spending your afternoon with us, and hopefully you have a good rest of your day.
Thank you so much. I really appreciate it.
Thank you so much. Appreciate it. All right, everyone. Thank you so much. We'll see you next week. Bye now.